Welcome to another lecture by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 12, Cancer Biology. So in this lecture, we will briefly discuss the hallmarks of cancer. We will discuss oncogenes and tumor suppressor genes. We will compare and differentiate between angiogenesis and metastasis, or metastasis. We will also look at molecular basis for cancer therapies and end with a summary. Now, let's discuss the hallmarks of cancer. So, cancer cells exhibit several defining characteristics known as the hallmarks of cancer. First one is uncontrolled proliferation. So, it's ignoring signals to stop dividing. Second one is it's evading apoptosis, meaning it's avoiding programmed cell death. Remember, we had checkpoints in place during cell division, and if our systems recognize that a mutation has occurred that should not be happening, or a proliferation is going on that's not supposed to be happening, our body has mechanisms in place to program the cell to die, the faulty cell to die. But one hallmark of cancer is that it's evading this system. So this uncontrolled proliferation can just continue. Furthermore, as this continues, we have angiogenesis, and this stimulates blood vessel growth to supply the tumor. As this tumor is growing, it needs blood supply, and it's creating blood vessels to grow to supply it with its nutrients. Number 4, immortality, so it can divide indefinitely by maintaining telomeres. And when things become very serious, we talk about invasion and metastasis, which means it's spreading to distant tissues. It's invading to other areas of our body. This is when we say that the cancer has spread. And finally, immune evasion. So it avoids being detected and destroyed by our immune system. So it's not only avoiding cell programmed cell death on this cellular level, but it's also avoiding our immune system from detecting it and destroying it. Now, oncogenes and tumor suppressor genes. Oncogenes, their normal function is to promote cell growth as proto-oncogenes. However, when they are mutated, they become overactive, leading to excessive cell division. Examples of these oncogenes are the RAS gene or the MyC gene or MyC gene. We then have tumor suppressor genes. Their normal function is to halt cell division or promote reparation or repair. If it's mutated, it loses its function, so it allows for unchecked growth. Examples of tumor suppressor genes are p53 or RB1. And then we have DNA repair genes. Their normal function is to fix DNA damage. Mutations can lead to genomic instability. So oncogenes are like a gas pedal stuck down, while tumor suppressor genes are like brakes that have failed. So it's quite fascinating how our body normally have these systems in place. But why is medicine a subject of study? Why do we need doctors? Because these systems that normally promote, for example, oncogenes, normally promote cell growth. That's a positive thing. And we call them proto-oncogenes. But if they become overactive, we will have uncontrolled cell division happening, or excessive cell division. Luckily, why we're not developing cancer in even bigger numbers is because we also have tumor suppressor genes. And they are there to stop faulty genes, faulty cells from dividing. Or if they are damaged, they promote reparation. So these are quite delicate and fascinating systems to study. Now, let's explore how tumors grow and spread to other parts of the body. So we mentioned in the hallmarks of cancer, angiogenesis and metastasis. So angiogenesis is blood vessel formation. Now these tumors that are growing secrete signals, and these signals asks essentially for new blood vessels to grow because it needs oxygen and nutrients to keep growing. Now it sounds like a nice thing, something growing and it's supplying itself to grow. But we're talking about a tumor, right? Then we have metastasis, or when we say that the tumor or the cancer has metastasized, we are saying that it has spread now to other tissues. So the cancer cells break away from the primary tumor. So if we say we differentiate between a benign tumor, it has not spread, and then we have metastasis, or when we say that the tumor has metastasized, part of the cancer cells break away. And what they do is they invade the extracellular matrix and travel through the bloodstream or the lymphatic system elsewhere. And what they do then is they will form secondary tumors in other distant organs and areas. So to give an illustration, angiogenesis is like building roads to supply a growing city. And metastasis or metastasis is like invaders traveling to conquer new territories. So let's examine how our understanding of cancer biology has led to targeted therapies. Now, targeted therapies are drugs designed to block specific molecules that are driving cancer. Examples of these include, for example, HER2 inhibitors or HER2 inhibitors for breast cancer, EGFR inhibitors for lung cancer. What about immunotherapy? Well, enhancing the immune system's ability to detect and destroy cancer cells. Examples could be checkpoint inhibitors like anti-PD-1 or anti-CTLA-4. Another is angiogenesis inhibitors, where we block blood vessel growth to starve the tumor. Remember, once the tumor begins to grow enough, it now needs blood supply to get nutrients to continue growing. But what we do is we block this from happening. So we let the tumor starve. We also have gene therapy, so correcting or replacing defective genes involved in cancer. So in summary then, cancer arises from genetic mutations affecting oncogenes, tumor suppressor genes, and DNA repair genes. Hallmarks of cancer include uncontrolled growth, immortality, and the ability to invade and metastasize. Also avoiding and evading the immune system and evading programmed cell death are also hallmarks of cancer. So understanding cancer biology has led to advanced therapies like targeted drugs and immunotherapy. And that's the end of chapter 12. Continue now to chapter 13.